I'm David Koslicki, Associate Professor at Penn State University. I always find it a challenge to give consecutive CGSI talks because the faculty usually stays the same, but the trainees all change. And so apologies to the faculty, but you're welcome to the students. I'm still going to go over all the main definitions and stuff like that that I did last year when I gave a similar talk to this. So today we're going to talk about sketching for metagenomic analysis. So the plan is to talk about sketching in general, move to metagenomics, and then talk about two specific applications with taxonomic profiling and functional profiling. I had wanted to talk about a whole lot more in sketching, but 45 minutes is 45 minutes. So let's start with sketching. What is sketching and approximate data structures and randomized algorithms, et cetera? What are all these things collectively? Well, these are approaches and data structures that trade off accuracy for efficiency. So basically, when you don't need an exactly precise answer, you're allowed to have a little bit of wiggle room and bound on that wiggle room of your estimate. And that's what these kinds of approaches give you. And honestly, in biology, close enough is often good enough. If a percent change of 1% versus 1.001% doesn't often make or break a certain case. And these sorts of approaches have a lot of different applications, and you can slice and dice them in different ways. Here I'll do it by query type. So you could be interested in, for example, membership queries, things like Bloom filters, cuckoo filters, et cetera, that answer questions like, does this k-mer or does this sequence appear in this particular genome or this particular sequencing set? You can also do things like cardinality estimates. So how many distinct k-mers do I have in my set, or how many distinct reads do I have in my sample? And you can use things like hyperlog to do this. Frequency and heavy hitter information, what are my most frequent stuff? Similarity and distance measurements, how similar are these two sequencing sets or two genomes based off of ANI, which we heard from Czar a bit earlier about using ANI. And nearest neighbor search, quantile, order statistics, et cetera, a whole bunch of different applications. So what we're going to do in this talk is focus primarily on the similarity and distance measurements. So things about like how similar or how different are these sequencing sets. One note, because we've already heard a little bit about sketching, and thank you, Czar, for talking about minimizers earlier, because I wanted to draw a distinction between two different ways these sort of sketching approaches are applied in computational biology. So one perspective is sketching k-mers as sets. The other perspective is sketching k-mers as a linear sequence. And it's subtle but distinct because one approach, particularly the one on the left, completely destroys linearity location information, whereas the second approach tries to preserve it. So the first approach basically takes all your reads and then turns it and gets all the k-mers and forgets all the information about which k-mer came from which read and which part of which read. And then from there, you might do something like randomly sample, and that random sample is then going to be your sketch. And critically, it's going to depend on how you randomly sample. In contrast, sketching k-mers as a linear sequence, you have your read or your genome or whatever, and you look at all these k-mers here, and you take a sliding window. And you take this window and you move it across your sequence, and in there you select some special k-mers. Special k-mers that are either lexicographically small or small according to some hash function. And then these special k-mers become your minimizer sketch. Sometimes, but not always, and it's not been completely settled or even looked at in the literature, is when can you take a minimizer sketch and treat it like this and it still has the nice properties, and conversely. But in any case, for this talk, I'm going to talk about the first one. I'm counting or treating k-mers as just a bag of k-mers, ignoring order information. Okay, so back to these similarity and distance estimates sketching approaches. Let's do a warm-up with MinHash. So I'm assuming that some fraction of you have heard of MinHash before. I'm just curious. Raise your hand if you've heard of MinHash. Good, not everybody raised their hand, so this next slide will be helpful. So this is a classic sketching technique defined by Broder back when he was with AltaVista, and AltaVista was a thing. And it hinges on a hash function. And so this hash function here, you can think about it as mapping k-mers to uniformly random numbers. So you can take a k-mer like ACTGC here, and it assigns a number 7. Take a different k-mer, it assigns a different number. The way that MinHash works is you would take your reads, and you start streaming the k-mers through. You take each one of these k-mers, and you apply your hash function h to get what random number has been assigned to it. You get these numbers, and then you select either the largest or smallest. Here we'll say smallest, n of these numbers here, and then that turns out to be your bottom n sketch. So you have selected a fixed amount, n, of these numbers that came from your individual k-mers. And so if your hash function is bijective, or even just injective, you have that these correspond to unique k-mers over here, so you can think about selecting random k-mers. The key idea of this approach is that if in one set A you have a k-mer that made it into your bottom k sketch, the random number associated with it is small. And if that k-mer also appeared in a different set B, then with pretty high likelihood it's also going to make it into the sketch. And because of this, you can measure similarity between these sets of k-mers, particularly with things like Jacquard or containment index. So recall that Jacquard index is the size of the intersection of two sets divided by the size of the union. And you can prove that averaging over the different hash functions that you use, the Jacquard index of the bottom n sketch of these two sets is the same as the Jacquard index of the original two sets. And the really nifty thing is that your original sets A and B can have billions or trillions of elements, and you have subsampled just n of them. And so you can control that n to be, well, how much RAM or disk space or whatever I have to be able to facilitate this estimate. And again, I said there's some sort of provable guarantee that your estimates aren't too far off. And you end up with equations like this that you do not have to read, but it basically says that the likelihood that your estimate deviates by a large amount from the thing you're trying to estimate is small. So it's unlikely that you'll have a bad estimate. Cool, that's fantastic. MinHash is great. It's been applied in things like MASH, MashMap, etc. But unfortunately, it's not so good for sets of unbalanced size. So for example, if you have on one side a big metagenome, and on the other side a little viral genome, then, well, let's think about what happens. If we take 17 points from this very large set of k-mers and 17 points from this smaller set of k-mers, notice that in essence I have sampled this set more deeply than the other one over here. And this error right here is controlled quite a bit by how deeply you sample this bigger set. So MinHash, not so good when your sets are a very different size. So this motivated another kind of sketching technique called FracMinHash. The basic idea here is instead of sampling a fixed number of k-mers, we can do a fraction of k-mers. So pick a fraction, so like some scale factor s between 0 and 1, that's going to be how much compression you want to do. So if you pick an s of 1, that's I'm doing no sketching, I sample everything. A smaller one, I'm selecting a smaller fraction. Again, we have a hash function, maps our k-mers to numbers, and then a FracMinHash sketch, which really interestingly, like a whole lot of people came up with this idea kind of simultaneously. These are called things like min-code syncmers and scaled MinHashes and FracMinHashes and everything. And so we all got together on when it was called Twitter and sort of hashed out what we were going to call this thing. Yeah, that was actually unintentional. But in any case, so a FracMinHash sketch is defined as follows. So a FracMinHash sketch of your set A, you're going to take those hash values that are below some fraction of your maximum hash value. So as a pictorial representation of this, we have our set of k-mers, A, floating around, and we have our number line that represents the output of my hash function, here going from 0 to 100. So my capital H is 100. And I'm going to pick some scaled factor, I'm going to say 20%. And then if you take a uniformly random injective hash function, then it maps these k-mers to these unique numbers over here, and it keeps mapping, 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 and then all you do is retain the ones that have fallen below your threshold. And it's pretty easy to see that then you will have sampled uniformly randomly S% of the k-mers in your original set. And that's your sketch. You can then adjust the scale factor up and down, once again, depending on how much RAM and disk space you have and how accurate you want to be. So what does this get you in comparison to MinHash? A much tighter estimate. So take a big sequencing set, take a genome, put C fraction of that genome into your sequencing set, use MinHash, use FracMinHash to try to recover that C percentage of the genome in the big sequencing set, and you get a picture like this. On the x-axis is what the true containment index is, how much that genome was in your big sequencing set, and over here is the computed one. And you can see that the MinHash estimate has pretty wide variance in comparison to the FracMinHash, which is much closer, even though they both have the same expected value. Great, so with unbalanced set sizes, FracMinHash is good. But so far as I've introduced it, these kind of approaches kind of divorced from the biology, because yeah, okay, we can get containment index or Jacquard index from it, but how can I connect it to biology? Well, we've already heard of average nucleotide identity, this ANI, but let's recap the definition real quick. This is the ratio of matching versus all characters in two aligned sequences. So let's say we have two sequences, A and B. I align them, and I have highlighted in red where they differ. So if you count, you'll notice that there's 15 total nucleotides, and 2 of them don't match. So that means 13 of them do match. So the ANI here is 13 over 15. Well, it turns out that these Jacquard and containment indices can be related to average nucleotide identity. And it's really simple if you assume a substitution-only model. So let's just do that real quickly. Assume we only have substitutions, no insertions or deletions. ANI is very popular because lots of studies have shown that for organisms that are like the same species or same strain, they're going to uniformly have very high ANI with each other, whereas things that are more distantly related have low ANI. So things of different species, lower ANI, same species, higher ANI. So it's very biologically useful to distinguish relatedness of different organisms. Okay, but let's go back and connect this to k-mers. Now I'm going to refer to A and B as the k-mers in these individual sequences. So what I'm going to do is I'm going to relate containment index to ANI real quickly. So containment index is how much of A is contained in B here. Well, if we're considering 5-mers, notice all the 5-mers in this region right here that don't have a mutation, they're exactly the same. Those things in the middle correspond to the stuff in the overlap. However, every time I have a k-mer that overlaps one of these mutations, if my k-mer size is big enough, that k-mer in B is going to be different than the k-mer in A. Therefore, resulting in one k-mer in the A side of the symmetric difference and in the B side of the symmetric difference. So to compute my containment index, I'm basically taking all the k-mers in A, and I'm removing all the k-mers that have been mutated. Because every time I have a mutated k-mer, it gets, boop, kicked out of A over here into B. Divide by the number of k-mers in A, and that's this quantity right here, and you'll notice that, oh wait, this is 1 minus the fraction of mutated k-mers. Well, that's just like the other way around of ANI, right? The fraction of matching k-mers. So I take 1 minus that quantity, and I get ANI. Okay, sweet. So I can now relate this containment index to a biologically relevant quantity, and I can use these hashing techniques to estimate this containment index. And it turns out you can do this with a lot of different biological quantities. So you can use FracMinHash sketches, and to some extent MinHash sketches, to do ANI estimation, like we just said. And you can actually extend that model from a substitution only into a substitution, insertion, and deletion. That's coming out in WABI in a couple months. If you're interested, the paper's also on bioRxiv. But it allows you to do things like other metrics, cosine similarity, Manhattan distance, stuff like this can also be estimated with FracMinHash sketches. You can do metagenomic taxonomic profiling, functional profiling. You can estimate DNDS. So DNDS, you might know there's a ratio of non-synonymous versus synonymous mutations. Gives you an idea of evolutionary pressure, if it's positive, negative, or neutral selection. You can estimate those things using FracMinHash sketches. And there's a lot of like user-friendly tooling around this. So Titus Brown, for instance, has implemented very helpfully a lot of our theory algorithms and stuff like that into this nice package called SourMash. So for the next 30 minutes or so of the talk, we're going to drill down on these two, the taxonomic profiling and the functional profiling. Any questions before we transition to this? Yeah?
Just a quick question. Then where you have the ANI versus contamination, the number of mutated chimeras and the number of mutations is different, but you need 1 minus d to the power of k.
Yes, right. So I have not given you what the formula is for this. Yep, and it's 1 minus p to the power k. Yeah, exactly. Yep. Is that a hand? Cool. Okay, so what is this metagenomics? I'm assuming here that not everybody is intimately familiar with metagenomics, so quick 30-second recap. It's a study of microbial communities through their sampled DNA. Oftentimes, and unfortunately, bacteria sort of dominates metagenomic studies, but let's not forget viruses, fungi, archaea. So why should you care? Microbes are ubiquitous. You're about as much bacteria as you are human when it comes to cell counts. And you're more bacteria if you count the diversity of different genes in the bacteria in and on you. And the reason why this is bacteria can be tiny, tiny. So human white blood cell, bunch of E. coli. You can fit a lot of these things in you. These are implicated for lots of diseases, critical to life. You wouldn't be able to function without microbes. So how do we study it? What's the data? Well, we take an environmental sample. You do a bunch of wet lab wizardry, and you stick it onto one of these big machines that we saw, and at an extremely high level, pretend these are genomes floating around in your sample. They all get shredded up, and then at the end, you end up with tens of gigabytes to terabytes worth of these reads. Currently, in metagenomics, short-read technology still dominates because depth is king here when your communities are very diverse. Because when you've got thousands of different species in a sample, you need to sample very deeply to be able to get even 1x coverage on each one of those members. Long-read technology is starting to really take off because at least for the heavy hitters, the things that are of high abundance, you can basically, with long-read technology, read out the entire genome, which is super handy for some applications. But for here, I'm just going to consider short-read technology. With these short reads, you can do a lot of different things. You can do taxonomic profiling. What organisms or what taxa are in my sample and at what relative abundance. You can try to put these genomes back together with assembly, and then after you do assembly, you get a bunch of contigs, and you might want to say, well, can I group these contigs into things that I think are the same organism? And that's going to be genome binning, or can I group these contigs into individual taxa? Slightly different thing called taxonomic binning. You can also take 2 metagenome samples just directly and see, how similar or different are they? If I take it in 2 different spatial locations, how similar or different are my communities? And then, what functions are these metagenomic communities capable of doing? And that would be functional profiling. As I said, we're gonna, there you go, now you can take the picture. We're gonna focus on taxonomic profiling and functional profiling primarily. Okay, so taxonomic profiling. Well, if you work in metagenomics, you know there are dozens and dozens of different taxonomic profiling tools. We already heard Czar talk about Kraken. That one is used ubiquitously along with things like Metaflan and Motus, and I could go on and on. But there's an independent consortium that tries to assess metagenomic computational tools, in particular using unpublished mixed in with public genomes, called the CAMI consortium. And in their benchmarking, they have found that performance of these different tools vary extremely wildly. So the way that this approach works is that this is a competition. The consortium puts out the test data sets and say, run your tools, give us the results, we're going to hide the ground truth, and then we will evaluate them independently. And we see that basically you get stuff like this. Here are a bunch of different tools to do taxonomic profiling. And here is purity versus completeness, or precision versus recall. You can see it's kind of like a scatter shot. It's just a scatter shot. It's all over the place, right? You have some tools that are really precise and terribly insensitive, and the other way around. It's kind of all over the place. And that's just a single sample. If you do this in different environments, this one was marine, it can look completely different. And some of the tools that are the most frequently used oftentimes have unexpected behaviors. Like here, this is Treangen on one of the plant-associated metagenomes. Here again, we have purity or precision, completeness or recall. And notice that the purity is real low, just about 0. Oftentimes lights up your database. But if you throw away the low abundance ones, that 1% filtering right here, then your purity goes up, but your completeness just dives down. So and this actually sort of motivates why people should not use Treangen for bacterial taxonomic profiling. That's what Bracken is for, right? And that's built on top of Treangen. But even then, as we saw, Bracken in this case right here, our best tool would be up right over here, so there is still room for improvement in these. Okay, so let's step back a minute and consider the most simple problem. Instead of taxonomic profiling, let's do genome presence-absence. From these reads, I have some sort of reference database with, I don't know, a couple million different genomes inside of it, and these are going to be my microbial genomes. I want to go through and just say, is this genome in my sample or not? And so I want like a 0-1 vector saying, yes, this genome is in my sample, no, that genome is not, et cetera, et cetera. But as we just sort of saw with Treangen, there's oftentimes a big problem with trying to disentangle the low abundance organisms from the noise. So a prototypical plot of a taxonomic profile of a metagenome looks like this. You have some highly abundant organisms and a big old long tail of low abundance organisms that gets mixed up with the noise of whatever computational technique that you're using. And so current approaches to address this are oftentimes just hard thresholding. Let's take some arbitrary 1% or whatever and just ignore everything below that. It's one of the most popular ways to deal with this problem. It throws out a lot of true positives, but it does help you reduce your false positives. Okay, so I want to talk about a FracMinHash based taxonomic profiler that was developed in my lab called YACHT. With a PhD student that I was working on with this, most of the other computational techniques in his thesis were nautical themed. And so he was like, can we backronym it from YACHT? So it's yes, no answers for community hypothesis testing. So give it a few inputs, give me a confidence value, like 95%, some desired false positive rate. What percentage ANI means in my sample, right? Because your reference genomes are never going to be exactly represented in your sample. There's always going to be either sequencing noise or evolutionary divergence. So, 95% ANI, is that good enough for you, saying it's the same species? Or do you want 99.995 ANI, you want the same strain? And then, what percentage of that genome do you have to see to be able to say, yes, it's in my sample? I.e., what coverage do you want to desire? Given those 4 inputs, YACHT will tell you if there's enough evidence in the terms of k-mers to support that reference genome being in your sample. So basically, we don't have to do this sort of hard thresholding stuff. We can go one by one with each one and ask, have we seen enough evidence in the form of these k-mers to support the hypothesis that is present in my sample? The actual algorithm is not even 10 lines long, and at the heart of it is this little simple statistical test that we'll talk about in a minute. So how does it work? You take your reads, you take your reference database, you convert everything into k-mers. That would be a big headache because that'd be a lot of k-mers, so you actually subsample it and you take FracMinHash sketches of all these things. And then you only retain the reference database entries that have a non-zero overlap with your sample. Because if there's no overlap, there's no evidence at all that it's in your sample. You can just completely ignore it. And then, and this is kind of a key thing, this is taking inspiration from Treangen, is you look at the genome-specific k-mers. Of the ones that do overlap with your sample, which k-mers are unique to the genomes? So if you see that k-mer, you have more confidence that it's present in your sample. And then you calculate if there's enough support, you've seen enough of those, to support the claim that that genome is in your sample. What do I mean enough support? Well, it's that test, which we'll talk about right now. The hypothesis test has a null hypothesis that is kind of weird. The null hypothesis is the genome is present in the sample, kind of the opposite way around. And that's really the only way that we can avoid a sort of generative model of trying to say, what does it look like to have an arbitrary genome be in my sample at a certain ANI at a certain coverage. So we have our genome, our null hypothesis, we take all of our k-mers, and all those k-mers are in my sample. So with that null hypothesis here, however, we have evolutionary divergence and error. So we're going to have a mutated version of this genome. And any little mutation is going to affect those k-mers around that mutation. But remember, we're sketching this, so we're not considering all the k-mers, we're subsampling to just look at the FracMinHash sketched k-mers here. And if you notice, now I actually just have a binomial distribution. Each one of these k-mers are either in my sample or they're not in my sample. So the number of k-mers shared between this reference genome that's been mutated and my sample is going to behave according to a binomial distribution, which has a parameter associated with ANI. And then all you have to do is you pick and say, what is the fewest k-mers that I would expect to see if that mutated genome was present? You can easily calculate that. And then your test is simply say, well, let's go look and see how many k-mers I did see. If it's more than the minimum amount, then hey, the genome's in the sample, I accept the null hypothesis. If I haven't seen enough k-mers, then nope, it's not in the sample. And the nice thing is this gives you a controllable false negative rate. So if you didn't follow that, the takeaway is it's just a way to say, did I see enough k-mers to say it's in my sample? With this, there are a few knobs that you can twiddle. As always, don't ever just run bioinformatics tools straight after downloading them. Understand the parameters and the impact of those parameters on your interpretations thereof. The ANI threshold is pretty important because that controls, the amount of evolutionary divergence between your reference genomes and the things actually in your sample. Your significance, your probability of your negatives being true negatives, and this min coverage is also important. Of those genomes that are unique, of those k-mers that are unique to each one of these genomes, how many of them do you need to see to say that it's present in my sample? Just one? Maybe a handful? That's really what the user has to set. So, knowing that those are the two most important parameters, what does it look like on this CAMI test data here? So here's a rhizosphere sample, just a few different tools, and then these are versions of YACHT as I move my minimum coverage from very large to small. And you can see that as you move your minimum coverage down to lower and lower, you have to see fewer and fewer fraction of those unique k-mers to say that's in my sample. Of course, you would say that the sensitivity is going to go up, but your precision is going to drop. And conversely, as you move it the other way. But at least it's nice that all of these are up or further to the right than this. But of course, you change the environment and things will change. So for example, if now I move to marine samples, then at a few different parameter settings, it might be more precise, but less sensitive. Turns out centrifuge in this case, actually centrifuge in this case does kind of the better overall, but at least this curve as you move your minimum coverage is nice and up and to the right. So same thing happens as you change environment. So takeaway from this, one slide that I didn't add is that this is ridiculously quick. You're talking like a second or two to do your taxonomic profile of an entire metagenome. You're talking about maybe 10, 15, 30 seconds to do your sketching of this. So it's extremely quick in comparison, especially to things like Treangen, which is already super fast. Okay, taxonomic profiling questions before we move on to functional? Yeah?
How do you account for the horizontally transferred elements?
Yes, how do we account for horizontally transferred elements? So you are in luck if it turns out that the organism from which that gene got transferred also has non-zero overlap with your sample, because that region, since it's shared between more than one genome, you aren't going to consider k-mers from there as being indicative of one or the other. We're only looking at genome-specific k-mers. Yep. If you are unlucky and that reference has zero overlap with your sample because you just happen not to sequence that gene, then yeah, that's probably why you end up with stuff down here instead of up there. Yep. You can play the same game that we just did with functional profiling, with a slight modification. So functional profiling is after you sequence your environment, I want to know what genes, what functions, what gene orthologs, et cetera, are in my metagenome. So what's the functional potential of my metagenome? Overwhelmingly, the approaches that are used nowadays are alignment-based. So Diamond is the one that I see used the most to do this kind of functional annotation. This can get prohibitively expensive as your reference databases continue to grow and grow and grow. Diamond is ridiculously fast for what it does, but the inevitable march of the size of these databases make it harder and harder to apply. So it's perfect for these sketching-based approaches, which are orders of magnitude faster. Okay, well, if I'm doing functional annotation, I have to tell you what sort of function I'm annotating against. So I'm going to talk about KEGG. So we just chose because we had access to it at the time, and also because it's associated with a knowledge graph that I work with, to use KEGG, which KEGG KOs, or orthologous groups of proteins that are nice and hierarchically related, so that each one of these represents a set of orthologous amino acid sequences. This is nice because it connects to pathways and various other things like this. And a sort of pro tip, sketching genes is not a good idea. Genes are relatively small. If you're dealing with relatively small stuff, you should not be sketching because you're throwing away even more information. You should only sketch if you're dealing with very large sets. These sets of orthologs are large enough that it makes sense to sketch them. Don't sketch individual genes. So what's the pipeline of this FracMinHash-based functional profiler? That's a mouthful. Well, you take all these orthologs, here represented ortholog 1, 2, et cetera, and you sketch each ortholog individually and get those sketches. You take your sample, you also sketch it, and then you basically just run this nice little thing called sour mash prefetch, which does kind of a set cover problem where it's trying to say, okay, of all these k-mers here, which one of my orthologs has most of the hashes that overlap with it? Okay, assign those hashes to the next one, keep on repeating, and then at the end of the day, you get abundances associated with each one of these orthologs. So how does this work in practice? Well, as I've been saying, these sketching-based approaches are significantly faster than traditional exact approaches. So in this case, notice this is log seconds on the y-axis here. It's about 2 orders of magnitude faster than Diamond, this in its fast mode, which is still extremely fast. So basically, you're basically talking about hours versus a minute or so, minute or two. Also uses a whole lot less RAM because you're sketching things. You're taking a smaller fraction of your actual data. You have half an order of magnitude less RAM. Interestingly, it turns out to be very precise, even more precise than Diamond itself. So here, this is all on simulated metagenomes. This is an error rate, my sequencing error rate. This is our purity. And you notice that using a couple of different K sizes, these are amino acid k-mer sizes. The approach of this FMH fund profiler is more precise than Diamond down here. But of course, there's no free lunch. It's not as sensitive, which makes sense because you're doing a sketching approach. You're throwing away some information, so you're going to miss out on a few things. So for example, here again, error rate on this side is completeness. Diamond is wonderfully complete, gets 100%, but it's not super precise. But on the flip side, we get about a 20% performance hit in our completeness. So basically, this functional profiler is great for high-level general idea of large data sets. And then from there, you might then want to zoom in using something like Diamond to do a more refined analysis on fewer samples. The thing that sort of stuck out to us about this is that the actual ortholog relative abundances is more accurate than Diamond. And I would have thought that alignment-based technique would be more accurate. But it turns out there's just a lot of variance associated with Diamond in its estimate of the relative abundance of these KOs. And so the Pearson correlation, higher is better with a FracMinHash-based approach, and the Bray-Curtis distance, lower is better here as well. And it just turns out Diamond has high variance. This is probably due to HGT or some other similarities and things like that, that reads that could be associated with multiple different ones is, randomly chosen for other ones. Okay, so awesome. We still have 10 minutes for questions or an early break for lunch. Conclusion, sketching. It's uniquely well suited for genomic analysis because oftentimes we don't need an exact answer. We just need an answer that's close enough with some sort of guarantee about how good enough it is. And particularly for metagenomics, when we have these very large data sets, it's very well suited. You can connect these things to a lot of different biological quantities. My lab is very mathy, and I've tried to completely gloss over all of the math. But for each one of these things that I said you can connect it to, there are theorems guaranteeing, okay, exactly what your compression factor is and exactly how good your estimate is, et cetera. And if you're interested, you can go and grab these tools off of GitHub or Bioconda right now and run them, both Yacht and the functional profiler. And one thing that we're doing collaborating with Ryan is basically reanalyzing all of the metagenomes in the SRA using this uniform taxonomic and functional profiler. You might ask, well, I thought the SRA already gave you taxonomic profilers or profiles. Well, yes, but they're MinHash based. We already saw that MinHash might not be the best. So our first analysis is going to be how good are those taxonomic profilers or profiles that the SRA gives you? And then a whole bunch of exploratory analysis as well. If you've seen talks from me in past years, I also do a lot of knowledge graph stuff. And after we take all of these taxonomic and functional profiles, we can decorate them on knowledge graphs and maybe extract some additional insight. So these three are co-authors on the Yacht paper, and most everybody in the lab was a co-author on the FracMinHash functional profiler. This is not an exhaustive picture, but I don't actually had a picture of my entire group at the same time. And thank you to funding agencies for making this possible. Thank you for your attention.